All right, hi everyone. Thank you very much, Matt, for the introduction and Davor for hosting. I also want to say thank you to JMCC for inviting me to do this webinar. It really is an honour and a privilege to be able to share my work with a wider audience on this platform. I'd also just like to say again, thank you for handling our publication and for the editorial on our work. So today, the title of our paper is Standardized Method for Cardiomyocyte Isolation and Purification from Individual Neonatal Infant and Adult Hearts. But first, I'd like to acknowledge the Indigenous peoples of Australia, who are the Aboriginal and Torres Strait Islander people, who are the traditional custodians of the land for more than 60,000 years on which we live, work and meet. And on the land of the Victor Chang Cardiac Research Institute in Darlinghurst, I pay my respects to the Gadigal people of the Eora Nation, to their elders past, present and emerging, and also of the Dharawal people based in South Sydney, where I'm speaking from today. So back to the work. This protocol was really developed to study postnatal cardiac growth and cardiomyocyte development, which has really exploded in the last 10 years to look at maturation. And so one of the benefits of doing this is to look at the pathogenesis that occurs with heart development and cardiomyocyte maturation. And those include the maturation defects that occur with, that are caused by congenital heart disease or cardiomyopathies, or with acquired heart diseases. And I take this thought further to say that the postnatal period really is a period that represents a therapeutic opportunity for the treatment of many types of cardiovascular diseases that occur in children and adults. Another usefulness for understanding postnatal development is to be able to define those innate molecular mechanisms for cell proliferation and differentiation, which are processes that we could potentially manipulate to stimulate cardiomyocyte regeneration as a repair mechanism. And again, this is a huge field and it's a really exciting one to be in. And finally, it's also relevant to the human pluripotent stem cell cardiomyocyte field, especially for understanding endogenous maturation to help mature these cells further, which are used for human diseases, modeling human diseases, drug testing and cell therapy. However, the model systems for understanding postnatal cardiomyocyte maturation are lacking. For example, human samples spanning different ages are a very limited resource, while cell lines are not presently adequate for this purpose. And so primary rodent cardiomyocytes have really been the main model used in this area, and these methods have been continually developed since the 1960s. However, the current limitations to these isolation methods are that they are age dependent, and this means that they differ considerably with respect to tissue digestion conditions, processing times, and the number of parts used. And there's a big disparity between the isolation procedures from the neonatal heart and the adult heart. And so I'll just quickly recap for those that are not familiar with this protocol. The traditional method for isolating neonatal cardiomyocytes is batch chopping, which uses 5 to 15 hearts that are digested with trypsin, usually at 4 degrees overnight. And this is quite different to adult cardiomyocyte isolations that are usually performed using Langendorf retrograde perfusion, where the proteolytic enzymes collagenase are perfused through the coronary system at 37 degrees. And this will take 12 to 15 minutes per heart. Now, there are commercial kits available that reduce the time taken for neonatal cardiomyocyte isolations to 1 hour. However, this is a lot more than that 12 to 15 minute mark in the adults using Langendorf perfusion. And then we have the ages in between that, where different labs have tried both methods. And so there isn't really a standardized method for these ages, but there is a greater need to be able to isolate these cardiomyocytes. And so this is just a picture overview of the batch chopping method, where hearts are pulled and they are chopped up into smaller chunks of tissue to increase the surface area for serial digestions overnight. And for those who are unfamiliar with the Langendorf diffusion methods, these are my go-to methods when I was learning about Langendorf and really trying to get a good understanding of how it works. So these are two different Langendorf setups that pump the proteolytic enzymes through the coronary system of the heart, shown here. And there are quite a few different parameters to consider when doing this, and this is just a comparison of those. So once we have our cardiac cell population for cell type specific studies, which is what our lab is interested in, we really wanted to purify the cardiomyocyte population. However, these protocols are also age dependent. And so for a neonatal cardiomyocyte purification method, the traditional way to do this is by differential plating. So that's purification by cell culture. And again, this is done overnight and using those isolated cells from 5 to 15 hertz. In comparison, adult cardiomyocytes can be enriched by centrifugation or gravity settling, sometimes also by Perkel gradients. They can also be purified by cell culture, and this will take around 3 hours. There are, again, commercial kits that have reduced the time taken for neonatal cardiomyocytes to be purified within an hour. And again, that kind of leaves the ages in between during this maturation phase unanswered for. And so there isn't really a standardized method for infant ages. And so our goal was to standardize cardiomyocyte isolation and purification from individual hearts at any postnatal age. And so we did this by adapting the Langendorf diffusion method for cardiomyocyte isolation. We've also introduced an immunomagnetic cell separation step to purify the cardiomyocytes by removing the non-myocytes using magnetic beads and collecting the unbound fraction of cardiomyocytes. We've then gone on to show that we can get enough RNA to look at sex-specific difference in the neonatal cardiomyocyte transcript chain. Further to this, we've developed a method to fix cardiomyocytes in situ. So this is directly after Langendorf diffusion with the coagulases. We will fuse the hearts with PFA, and this will retain the cytoarchitecture of the cells as they would be in the heart. So this is really good for cell morphology studies. So on this slide, I'm just showing an overview of our procedure, which takes around or just under an hour to isolate and purify the cardiomyocytes. So using those two kits, it would take you about 2 hours. And so we're really pleased that we're able to get this down to an hour for all ages. We also would simultaneously isolate cardiomyocytes from 2 hearts using 2 rigs at the same time. And so in a day, we'd probably isolate cardiomyocytes from a total of 8 hearts. So the first step of this procedure was the in situ cannulation of the heart, where we've developed an easier technique of in situ cannulation. So typically for the adult mouse heart, cannulations are performed ex vivo. And this is where the aorta is transected at the aortic arch. The heart is then excised and placed in ice cold buffer in a petri dish, and the aorta entrance to be identified, and then the short section of aorta is cannulated. However, with our method, what we do is transect the aorta at the descending aorta at the level of the diaphragm. This leaves a longer section that we can then use to guide onto the cannula. And this adaptation was really important for neonatal and infant hearts. And here you can see what our setup looks like. So this is a blunted needle used for the cannula attached to a 10 ml syringe. And you can see the relative size to a postnatal day 2 pup. We've also had to adapt our microsurgical tools for the different sizes. And here in panel C, you can see the differently sized aortas. At neonatal and infant ages, they're very delicate and more breakable. And then you can see what the hearts look like once they're cannulated and they're attached to the Langendorf rig here. This is an example of what our setup looks like. So one of the applications of this in situ cannulation technique is the potential to establish younger working heart models for contractility, pharmacological or ischemia reperfusion study, which are normally performed on adult hearts, but we're hoping this would be useful for understanding different diseases in younger hearts. So the second step to this method is the Langendorf retrograde perfusion once cannulated with the tissue digestion enzyme. And so we have adapted the flow rate for differently sized hearts. We use the same enzymes and we adjust the concentration of these enzymes per gram of body weight. And we've also, I've also adjusted the buffers to give us the same time taken per digestion. And so this is around 13 minutes at each age. And so the significance of this is that if we have the same method being used for different ages, that we can then have comparative studies investigating age-related changes using the same method. So the third part for those who don't do isolations is just to tease apart the digested heart. So you can see in this top panel, these tissues are teased apart with scissors and forceps. We then get this cardiac cell suspension, and you can start to see the cardiomyocytes pellet at the bottom of this tube. And this is what our preps look like for adult mouse cardiac cell isolation. So you can see the cardiomyocytes being the bigger rod-shaped cells and the non-myocytes dotted in between. And when we count the number of cardiomyocytes we get at each of these ages, for the neonatal heart, we get about 1.6 million cardiomyocytes, and the same for the infant ages. And then the adult heart is what you would typically see of around 2.2 million cardiomyocytes. And this has been really good to be able to benchmark these yields because there's not much information out there, especially for Langendorf perfusion, which we've developed for this purpose. Another thing that was really interesting is that the batch chopping method usually yields round cardiomyocytes. We actually found that 53% of our cells were rod-shaped, so they had retained their cell structure. And the rest of the preps were around 70% rod-shaped. So I then wanted to kind of see how does this compare to what other methods are yielding. So for example, one commercial kit yields 2 million cardiomyocytes per 100 mg of tissue, and that's using the 10 to 15 hearts per experiment. So in comparison, we're yielding a similar amount, but for one heart, neonatal heart, which is around 7 mg. This equates to around 22 million cardiomyocytes per 100 mg of tissue. So our protocol is yielding 11-fold more cardiomyocytes than this. These are seminal papers for the batch chopping method. I really enjoy reading these papers because you get a sense of the work that was required, and also all the details are provided for yield, which was really difficult to find when looking for information about neonatal and infant hearts. So these traditional batch chopping methods reported an extrapolated yield of about 0.6 million cardiomyocytes per heart, which is around 2.6-fold lower than using the Langendorf method. And improved methods have yielded similar to ours of about 1.3 million cardiomyocytes per heart. However, again, it does require multiple hearts to be used per isolation and took 12 to 16 hours. So the advantage of our protocol is that it enables individual replicates, where one heart provides a high number of cardiomyocytes. And this is done within 25 minutes. And because the yield is high, it reduces the animal usage required per experiment. So going back a step, we also developed a method to fix the cardiomyocytes in situ. So this is done by perfusing the hearts with PFA immediately after perfusing them with platinum. And by doing this, these cardiomyocytes retain their in vivo cytoarchitecture. And this was evidenced by there being 94% rod-shaped across all ages, as you can see in this graph on the left. And this is much higher than what you would get if you fix the cardiomyocytes post-isolation. So I think this really demonstrates that the Langdorf perfusion is a gentle method of tissue digestion, and it's the subsequent steps of tissue disaggregation and cell isolation that reduce the cell shape and result in cell death. And so we can use this method to also accurately assess the morphology of maturing proteins of cardiomyocytes. So in this figure, you can see these are neonatal cardiomyocytes from postnatal day 2, and they have these beautiful spindle-like structures where the ends taper off. And at the infant ages of postnatal day 10 and 13, these cells get much bigger, and again much bigger at day 70. What you can also see is how irregularly shaped they are. So not so much like the rod brick cells we see depicted in papers. So what I wanted to do was then measure the cell area, the 2D area by polarimetry, so outlining the cells to get the total area size, since these cells are so irregular. And over time, from neonatal to adult ages, the cell size increases by around 10-fold. And what was also interesting is that the cell size varies by a third at every single age in size. I also assessed the nucleation status at the different ages. So the majority of neonatal cardiomyocytes were mononucleated as expected, and the binucleation index increased to 70 to 80% at infant ages, which is reaching that status of 85% in the adult age. And I also looked at the heart weights. So these are heart weights taken from restricted litocytes that naturally occur from C57 black 6 mice of an average size of 6 to 8 pups, and plotted those against the cardiomyocyte area. And you can see they track very nicely together with age. And when plotted against one another, the cell size and the heart weight, I was surprised to see a perfect correlation score, but I guess unsurprisingly, the cell size is determining the heart weight and heart size. And then using this, so I should also say that I think the 2D cell area is a more sensitive measure of cell size. We often use width and length, and I think that's important for understanding physiological versus pathological hypertrophy. But in this case, it was quite clear that the 2D area was a very sensitive measure of cell size. And so using this in situ fixation method, we're also able to capture proliferating neonatal cardiomyocytes. And you can see, again, we have these beautiful spindle-shaped structures where the mid-body is positively stained with Aurora B kinase during active cytokinesis. And this has really only been shown in tissue sections or cells dividing in culture. So again, I think this indicates how gentle the Langdorf perfusion method is in combination with the PFA perfusion. So so in situ fixation is a valuable method for accurate studies of cell morphology. And I think this will be useful for detecting proteins of interest, as well as their spatial localization to better understand cell structure and function. So going back to the isolation and purification method, the next step in this process was to enrich cardiomyocytes. So this is normally done by gravity settling or by low-speed centrifugations. Regardless of which one you choose, the principle is the same. The larger cardiomyocytes will pellets, while the smaller non-myocytes remain in the supernatant, and they are removed with each wash. And so this is normally done for adult cardiac cell preparations, but we have adapted this for infant cardiomyocytes by increasing the speed. However, this method was omitted for neonatal cardiac cells since the cell sizes are similar and you don't get efficient separation. However, when we did this with the adult cardiomyocyte, although the majority of this field of view, if you look at this side, they generally look like they've been purified. And under a bright field microscope, these one clusters that are attached to cardiomyocytes are not clearly visible. But what I did notice were these strings of cells appearing. So when we stain them, you can see how many contaminating endothelial cells are still attached to cardiomyocytes after enrichment. And so that really prompted us to develop a method to remove these contaminating endothelial cells. So that led us to step 5, which is cardiomyocyte purification. And so to do this, I developed a method where we have the cardiac cell suspension here, and then pre-coated dynabeads with CD31 antibody, and add the cells and incubate them with the bees for 15 minutes at room temperature. I've just adapted this protocol. It's not what the cells look like. And then after 15 minutes, you can place the samples onto a magnetic rack, and this will remove the non-myocytes, and we can collect the unbound cardiomyocytes. And you can see the difference between these two fractions here, where there are a lot of contaminating endothelial cells in the antibody bead fraction. And we show that there are around 95% pure cardiomyocytes in the unbound fraction. So the advantage again of this is that it standardizes the removal of contaminating endothelial cells, as these may vary between preparations. Now we use a different method for the neonatal cardiomyocyte purification step. So this is by still by immunomagnetic cell separation, but using this kit from Miltenyi, where they have microbeads attached to a cocktail of proprietary antibodies against non-myocytes, leaving the cardiomyocytes. And when we use this ion matrix column and pass the sample through, the non-myocytes are retained in the column while the cardiomyocytes are in the flow through. And so generally this method appears to be quite gentle as well. It takes 15 minutes and we get these freshly purified cells. And this is an example of a really high quality preparation after immunomagnetic cell separation. And these cells are mounted on just on slides, and they're fresh, so they haven't been fixed or anything. And you can see that they're around 80 to 85% rod shape. And on the lower panel, there's a close-up of what these cells look like. Again, although we do see cell losses during the enrichment and purification steps, the cardiomyocyte yield was high. So for infant and neonatal hearts, we get around 0.8 to 1 million cells at the end of this process, and around 1.6 million in the adult cardiomyocyte. And some papers show that they are getting this after isolation, and we're showing that we get this after purification. But if we have more papers doing this kind of purification, hopefully they can use this information to benchmark what their procedure is doing in terms of yield. I should say the viability was either remained the same or was slightly reduced after purification. So we wanted to confirm that we did have pure cardiomyocytes, and the first was to assess this by immunocytochemistry, and we picked 3 different methods to show this. Okay, so for the immunocytochemistry, I'm showing the isolation fraction, the purification fraction, and the antibody bead bound fraction. And this is from the neonatal cardiomyocytes, secreted black 6 neonatal cardiomyocytes. So we have stained for cardiotropin T for cardiomyocytes, and IB4, I selected B4 for endothelial cells, and the DNA with topo. And then quantified the cells that we see across these fields of view for each cell type, and other cell type was categorized as being positive for the DNA stain, but not for the other two. And when we do this, we show that in the isolated fraction, we get around 60% cardiomyocytes, and this increases to around 96% in P2 hearts after purification. And you can see that in the antibody bead bound fraction, the majority of non-myocytes have been removed, which is evident in this picture. So the isolation, you can see a mixture, purification, a sea of beautiful green rod-shaped cells. And then in the antibody bead fraction, you can see all the endothelial cells that have been pulled out. And in the paper, we present a snapshot of this for the P10 cardiomyocytes enrichment and purification steps, where again, we're showing the isolation fraction below for each age, so 2, 10, 13, and adults. And after enrichment, the cardiomyocyte population increases about 15 to 20% at the infant ages, and it increases much more above 80% in the adult preparations. And then in the purification fractions, this averages to around 95% pure cardiomyocytes. And again, in the antibody B band fraction, a significant removal of non-myocytes at all ages. And this is quantifying around 1,500 to 2,000 cells per sample. So the second method to assess purity was by flow cytometry, which I wanted to do because we can then quantitatively look at 100,000 cells up to 500,000 cells per sample. And what you see here is on the y-axis, potassium iodide, which is staining all cells, and cardiotropin T, which is staining the cardiomyocytes. So the cardiomyocytes are present in the orange box on the right, and the non-myocytes in the purple box on the left. And you can appreciate that after purification, there's a clear separation between these two cell populations and a significant increase in the number of cardiomyocytes. And this averages out to around 96% pure cardiomyocytes at all ages. One of the technical limitations of using flow cytometry is for the intracellular staining of cardiotropin T, we fix the cells and use detergent washers. And I noticed that there was a bias towards larger cells, and in this analysis. However, we have used the same conditions for isolation and purification, and it's still showing a significant improvement compared to isolation. So the third method to assess cardiomyocyte purity was by qPCR. So this was quantifying cell type specific gene markers by endothelial cells being PCAM1, BWF, and fibroblasts, DDR2, and COL1A1. And when we look at the purified fraction versus the isolated fraction, these cell type gene markers are depleted. And in contrast, the cardiomyocyte markers post-purification was not significant. So there was no difference in the expression there. And so the goal for our work was to really do molecular study. So a lot of the protocols out there are developed for cell culture experiments or functional studies. However, we wanted freshly purified cardiomyocytes that we could isolate DNA, RNA, and protein from immediately. And so I've just documented here what our yield was for DNA, RNA, and protein, and normalized it to picogram per cell. So for example, if you had 1 million cells, you would have 7.6 micrograms of DNA from a neonatal purified cell population. And when you look at the RIN score, which indicates the quality of the RNA, you can see that the RIN score is really quite high, which is great to see that the quality of the RNA that's been extracted is good. And so, as I said before, this is mainly for molecular studies, such as multi-omic studies, which are, again, a hot topic right now to perform all of these, including the genomics, epigenomics, transcriptomics, and proteomics, as well as for carrying out the standard biochemical assays for PCR, qPCR, and Western blots. So the significance of this method is that it generates high-quality samples in sufficient yield from individual hearts that enable cell type-specific molecular studies. And indeed, we went on to do this ourselves to look at the sex differences in the neonatal cardiomyocyte transcriptome. So again, using litters with an average size of 6 to 8 pups, we took out 1 male, 1 female, and extracted or purified their cardiomyocytes and extracted RNA for sequencing. Before sequencing, we wanted to confirm by extracting the DNA from the same samples that we did indeed have the right sexes. So this is by amplifying the sex region located in the Y chromosome, which confirmed our male samples with the presence of a band at 400 to 500 base pairs in our male samples. They were absent in our female samples. And when we look at the body weight between males and females, there's no difference at postnatal day 2. But what we found is that there are around 13,000 genes expressed in the neonatal cardiomyocyte transcriptome. And when I perform the hierarchical clustering of these samples, you can see that they cluster together by the litter of origin. So litter 1, male and female here, litter 2, 4, 3, and 4 here. We also looked at the presence of non-myocytes in the RNA-seq data that we produced, and this shows that they're also significantly and much more lowly expressed in the cardiomyocyte markers as we expected. And this was looking at endothelial cell markers, fibroblast markers, vascular smooth muscle cell markers, and macrophages. And with the differential expression analysis, we found that there are 9 differentially expressed genes that are sex-specific, and they're also found on the XY chromosome. So out of the 13,000, there are only 9 that were differentially expressed in the neonatal cardiomyocyte transcriptome. And the biggest difference was the long coding RNA that's involved in exon activation exists, and this was highly expressed in female cardiomyocytes. So instead of me extracting the DNA, I then use this potentially as a marker to show that I confirm the sexes of my pups. But what this also highlights is that with the batch shopping method, one may use 8 hearts to produce 1 data point, and here we have 8 data points using this method. So in conclusion, we've established with you the Lowenthal method for a rapid and standardized procedure to isolate and purify cardiomyocytes, also using immunomagnetic cell separation. And they are in high yield, viability, and purity from mirroring hearts at any postnatal age. We've also developed an in situ fixation method to preserve this native cytoarchitecture of cardiomyocytes, which will be useful for accurately assessing cell morphology and proliferation, and also for use in immunocytochemistry experiments. And so the big benefit here is that by combining both of these methods, we can enable the molecular morphological profiling of high quality cardiomyocytes. And this is especially useful for younger hearts that can then be explored on an individual replicate basis, just as adult hearts are to evaluate genetic, therapeutic, surgical age, sex, and biological differences. And so in doing so, our data were the first to identify sex-specific genes that are expressed in the neonatal cardiomyocyte transcriptome. And altogether, these procedures, we think, represent a significant advance in the accessibility and application of primary cardiomyocytes as a model system, important for understanding cardiomyocyte biology, maturation, regeneration, diseases, and treatments for cardiovascular diseases. So with that, I would like to thank Bob Graham, who's been my mentor and supporting this work through funding discussion, as well as Dr. Siri Isma, who's also the corresponding author, and the contributions from the rest of the team: Michael, Andrea, Sarah, Ning, David, Paul, Nicola, Sam, and Azish. And finally, just say thank you again for tuning in, and I'm happy to take any questions.
Super. That was wonderful. Amy, it's great to read the paper, but it's even nicer to see you go through it. So that was wonderful. I think so. There's a couple of questions in the chat. So I think what we can do is we can maybe tackle those first, and then there's a few things I'm quite interested to talk about as well. So we can go for that afterwards. So regarding the questions, there's one thing that I think a lot of labs struggle with is having direct access to fresh tissue, especially in whole live animals. A lot of labs, they, for example, will have access to frozen tissue or fresh tissue, but not actually access to an animal facility at that time. So we do often get questions like this one, which is it possible to use techniques like this to actually take, for example, snap frozen tissue and isolate cardiomyocytes from that? I think it's quite difficult, but what do you think, Amy? Do you have any ideas on that?
So this is frozen human tissue?
It's frozen mouse tissue being referred to here, but I think the same would probably be true of frozen tissue as well.
I haven't personally tried it. I guess that's if you have a limited resource of tissue that you have only in the freezer. I know people extract nuclei from frozen tissues, but I think that's as far as what I've seen being performed. So I'm not too sure because I haven't done that myself.
Yeah, I think when we're talking about perfusion techniques, it's automatically going to be quite difficult to perfuse a chunk of frozen tissue, right? So perfusion, it comes in two flavors really, although I heard Miltony is making a commercial perfusion machine kit, so we'll watch this space for that. but yeah, as far as what you and I are doing, there's perfusion, retrograde perfusion via the aorta, and antegrade perfusion, which is what we usually do, which is basically pushing a needle into the ventricle space and injecting our buffers that way. Either of those.
I didn't mention antegrade perfusion because I thought you're the expert. You can talk to that rather than me.
Have you ever tried it actually?
I haven't.
Okay.
I haven't, and I should.
No, I think you have it working perfectly the way.
I don't think you should, Amy. I think you can have the results.
I think just stick with what you're doing.
But I think you can. The nice part about some of this work is that it doesn't really matter which perfusion protocol you use, because you could at least use other parts. For example, you could use antegrade perfusion and then the in situ fixation method, or purifying straight after the antegrade method using the immunomagnetic cell separation step. So hopefully there's other aspects that would be useful for multiple protocols.
We thought exactly that as well. There's a lot of complementary areas in here, so we're really keen to start trying in situ fixation using our method as well, because I think we'll get, for example, if we want to see hypertrophy in adult hearts after, for example, a TAC model or something where you expect to see hypertrophy, it's very difficult to measure hypertrophy in cardiomyocytes without fixing them, because they will tend to change and die. But this really, I think, will help to preserve the cytostructure, as you said.
And I think sections are also quite difficult, but there are definitely really interesting new protocols coming through that are able to visualize the whole cell in a section, especially for adult cardiomyocytes.
Yeah. I mean, coming back to this question, I think he's added some extra to it. So skipping. So I think so basically the question here is, say we have a piece of frozen tissue can we still perfuse it? I think perfusion is quite difficult because usually you're not having a whole heart. And actually when you freeze tissue, the cells are usually quite damaged. So these cells, the membranes are no longer intact. They'll have holes in them from ice crystals. I have seen a paper where they try and freeze tissue slowly in a cryopreservation medium using, for example, 10% DMSO. And then so the same way as you would freeze cells in culture, basically, and then revive them in the same way as well. And I have seen them say that cells retain viability after that. But even then, I think unless you have a whole piece of tissue, it's going to be quite difficult to perfuse that. And exactly what you were saying, Amy, about chunk methods, right? If you're trying to use a chunk method to digest heart tissue, your chance of getting viable myocytes is really very low, or even just intact myocytes is very low, because you really have to get your enzyme inside the tissue to kind of digest it from the inside out, right? If you try and digest it from the outside in, your viability and the number of cells you get is really quite small. Yeah. So I hope that would answer your question, Robert. It's something I think is going to be very difficult. Can you see the questions, Amy? Sorry.
Yes, I can.
Okay, you can. Yeah, this is in the chat actually.
Yeah, they're both in Q&A and chat. There's lots of questions.
Yeah.
Robert's happy.
Okay, great. But like you said, Amy, that says people who do this, they tend to do nuclei isolation, right? Which does work.
Yeah.
Yeah.
And
okay.
I think also what's interesting, just to add to that, when you're talking about neonatal cardiomyocytes and using the chunk method, it's interesting to me that the neonatal cardiomyocytes will all come out round shaped, but they're all viable. But if you try this with adults, none of them would be viable. But with the Langendorf perfusion method, only 53% retain their cell structure in the neonate. So they're less robust with the Langendorf in terms of keeping their cell structure, but more robust to be able to do the chunk method. So I'm not sure what's going on there, but yeah, interesting.
Absolutely. Yeah, I think they're a lot more plastic.
Yeah.
So there's another question here. So there's a question from Sarah Wood. It's asking asking about experience with isolated human cardiac tissue. So have you ever tried this, Amy? Have you ever had access to human tissue to try and dissociate?
No, I haven't. I know people have used Langendorf, for example, in rabbits and dogs, but no, I haven't personally used human tissue.
It's when you have such, I mean, generally when you get human tissue, you're not getting a whole heart.
No, it's a biopsy.
So yeah, you're going to get a biopsy. And I've heard that if you're very lucky, you'll get a big enough piece of biopsy which has maybe a blood vessel in it that you can actually cannulate on Langendorf, and
okay,
and you can actually digest it that way. But there is a paper, it's from a Chinese group, and they actually do something quite interesting with human heart tissue. They use a vibrating microtome, a vibrotome, they call it. What this does is it's able to cut very thin slices of the tissue, so you actually increase its surface area that way. And then you can put these slices directly into enzyme buffer. So it's kind of like chunk-based method, but using very finely sliced tissue. And the paper, it's quite convincing, I think, is that they get very nice looking human cardiomyocytes from that. So that I think that would maybe be the way to try it.
I guess, you know, probably one of the things that's worth discussing in this area or in this topic is that if you have a piece of human tissue or any tissue that's usually come from a diseased, you know, I don't know, a patient or whatever, or mouse, let's say there's an infarction, let's say there's, you know, there's already damage to some cells within that myocardium. Now, if you actually do that, if you're going to slice, then you're obviously going to cause more damage locally. And then I appreciate that, you know, obviously you isolate some cells, but are those representative of that heart anymore? Are these the healthiest cells that you can actually possibly get? Because we know that as soon as you get cells that are not healthy, they will die off very quickly in culture and during cell isolation protocols. So there is this question of, are you sort of biasing your findings through selecting healthier cells? I don't know what you two think of that, but, you know, would essentially slicing I don't know, a diseased heart, you know, with maybe a bit of ischemic areas or whatever, will it kill off those cells that are ischemic and only preserve the cells that are not? And therefore, is that really representative of the patient disease? What are your thoughts on this?
What do you think, Amy?
Well, I guess it depends if you're planning to resurrect the cells to use for something. Whereas I think a section, if you're going to image it frozen, that's fine. But if you're going to try and resurrect it from that state, especially a disease state, it might be difficult. I'm not even sure if the healthy tissue would survive that. So yeah, it would be interesting to see how that moves forward. But yeah, for now, I understand people want to kind of culture slices. That seems to work. I'm not sure. Somebody must have done it for diseased slices. But yeah, again, I'm not sure about that.
We cite this paper in the editorial, so I can't remember the authors' names right now, unfortunately. But if people are interested, then have a read of our editorial. And I think we do put it in there as an example of a method which is neither chunk nor perfusion. Now, I think what you're saying, Davor, is very true. I think that's also true of any isolation method, be it perfusion or chunk or not. You're going to tend to select for the healthier cells, and probably you select against the cells which are a little bit less healthy. So we haven't tried human tissue. We have tried pig atrial tissue. We didn't have a vibrotome, so we just tried to slice it manually, and it didn't work very well. But I think this was the importance of the vibrotome. Have you ever seen a vibrotome, Davor? How they work? Yeah, they're really nice. And I think the idea is that because the blade is very fine and it's vibrating, the pressure it exerts on the tissue is actually minimal. So you have to cut a certain width of slice, which I think is 200 microns. And when you do that, you hopefully preserve within the middle of that slice some cells which are not too damaged by that slicing procedure. And the images of the cells, they actually show them, and then they do look very good. So from what I can see, they look comparably healthy to the mice slice that we isolate. I haven't done this in my own hands, but yeah. The other thing is, I think that you have to work with what you've got, right? It would be there's not many people are going to give up their healthy heart for your research. So sometimes if it's a diseased heart, if you're lucky, you might have a surgeon who will take a biopsy for you from an area which they say is less diseased. Or if you're interested in disease, you can take the piece of tissue from that diseased area. How representative is that going to be? It's a question, but I think we're bound by these limitations.
I guess, I think you're right in saying that, but, I think Amy's work highlights a really important point. And I think you've seen this, Matt, as well in neonatal cardiomyocyte isolations, that, if you're too rough, you will get a different cell population. So obviously, Amy's detecting these kind of, sort of almost rectangular shaped cells in her neonatal preps. Now, historically, people don't see that, okay? And they still use these cells. Now, we all recognize that a cardiomyocyte should be rectangular, right? And that gives it its function, and therefore it's, it's probably highlights healthiness. Now, if we're destroying all these sort of cells that are supposed to be functional, what information can we derive from from our work? We've got to be quite careful. I think, Amy, you presented that really nicely and demonstrated that neonatal cells hearts have, these elongated cardiac myocytes. And if we've just worked on these little round shaped things, which do beat, then obviously we'll probably get the wrong conclusions. I don't know. I don't know what you think of that, but to me, that was quite a striking observation.
Yeah, I think from what I've read from previous studies, because the heart is the first organ to form and start beating in utero, those cardiomyocytes are actually spindle-shaped from a very early embryonic stage. And this has mainly been shown through sections in a very limited number of studies, but I think I've cited them in our paper. But really interesting to see, and not really talked about a lot, because I think the isolation has overtaken the sections in these spaces, and mostly you'll see round or star-shaped cells in cell culture. But the people who've been working on these sections know that they should be spindle-shaped. So what was cool to see for us was that we're able to get that in its isolated form, and even 94% being in that shape with the in-situ fixation method. So yeah, thank you. It was really exciting to see that we could retain that.
Sorry, go ahead, man.
That was what really jumped out at us, I think, when we saw the paper, was wow, these neonatal cells are really spindle shaped, which we haven't seen before. But I think what I would say, it really does depend on what you want to do with them. So for example, if you're going to look at protein and RNA, I wouldn't be surprised, and certainly DNA, I wouldn't be surprised if those don't necessarily change too much, whether the neonatal cell you get out is round or retains its spindle shape. With human cells, if you're interested in, for example, this patient has a genome genetic inherited disease, then if you can patch clamp a few of these cells and compare it to someone who doesn't, I think you could still probably glean something worthwhile from that. And certainly if you're looking at the RNA and DNA and things, then I think you could also get things useful from that. So I think it really depends on what you want to do with cells.
Yeah, good point. I'm wondering whether we should start answering these questions.
Let's keep going. So there's a question from Siavash. I think this one is, can you see these questions on the Q&A, Amy? So it says, thank you for the presentation. Do you perfuse the heart with collagenase and protease? If yes, for how long? I think maybe this is a case of reading the paper. You go into really great detail about the protocol and answer all of these questions. There.
Yeah.
There's one interesting part. Do you really stick to, so for example, in your paper you say how long you do these digestions for. Is there ever any situation where you think, oh, I need to finish this digestion early, it looks like it's done? Or do you think, oh, I think this looks like it needs more time, so I'm going to keep it going? For example, using here the example is using the color of the heart. So sometimes the heart gets pale, right, when it's digesting. So do you ever vary on that, or do you really stick quite closely to?
We stick quite closely to it. The only times where, if it hasn't worked for some reason or other, we would just increase the concentration of the enzymes rather than keep them going around, because whether you add 3 minutes or 2 minutes, it's not quite clear that we're going to get a more efficient digestion to us anyway. The heart kind of looks floppy after 10 minutes to us. So yeah, by eye, I can't say that we've got that kind of imperfect science sorted.
Yeah, it's certainly something that we do when we're doing the injection protocol. For whatever reason, sometimes we find maybe it's an older heart or it's a bit fibrotic, and we'll think it doesn't look like it's done yet. So we'll give it a few more rounds of enzyme until it does look pale and floppy. Then we'll stop then.
Yeah, certainly if it was a TAC heart or a disease model that had a significant fibrosis, we would probably consider doing that.
Okay, there's a question here from Honglin. Hi Amy, thank you for sharing. Could you remind the advantages of doing in situ cannulation compared to ex vivo cannulation? Is this method decreasing the time of pausing blood supply? The enzyme recipe, this is also something I was quite interested about. How do we choose the enzymes? And are we just relying too much on what people have done before, and rather than actually trying to optimize this ourselves?
Yeah, so both very good questions. I've thought about those questions a lot when developing this method. So the in situ cannulation, I learned off the Ming who was doing that method, and it was just so much easier even for adult cannulations. We don't need a microscope to cannulate the adult heart using that method. But for the neonates and infants, it was really important that we had enough of the aorta to be able to grip it and guide it onto the cannula. So the in situ was really good for that purpose. And in terms of comparing that to ex vivo, the difference is the temperature change. So in the thoracic cavity, we cannulate straight out of there. So I'm not sure how different that temperature change affects the heart, but we're really not cooling down the heart before we start to perfuse with this at 37 degrees. So I've wondered whether that makes a difference to the healthiness of our cells. But again, our adult cardiomyocytes are similar to what people see for ex vivo cannulation. So there's no real difference, I think, but for us it was just quicker to go straight from the thoracic cavity than to fiddle about in the petrogation and have the ice cold buffer. And the second question was the combinations. Yes, I would love to introduce some more ECM targeting enzymes for non-myocytes, and that might help get rid of that endothelial contamination that you see. So it'd be cool to combine and test multiple enzymes, but that obviously takes time. And I have really relied on previous papers to choose collagenase B and D combined with protease 14 for our digestions. Yeah.
And you really do, you very carefully adjust this by body weight of your
yes.
height, which we don't actually do. We are a bit more lazy than you are, so we just have a set recipe. How important do you think that is, that you really
again
to account?
We kind of adapted the Lounge-Young protocol that also adapt the concentrations to body weight. We did notice there was a threshold of body weight required for the neonatal and infant ages, which we've documented in our protocol. So they're not the actual body weights of the younger mice, they are for the adults, but we did need to reach a certain threshold where those concentrations were more effective. So yeah, that's just what we found to be useful, especially when you've got a combination of 3 enzymes versus people who use collagenase 2 and adjust it per mil.
I mean, while we're talking about this as well, some things is when we get questions about these people trying our protocol, the problems people I think are often coming up against is the choice of euthanasia. So whether we're using carbon dioxide, whether we're using isoflurane, whether we're using cervical dislocation, I think can make big changes. So what is your method of euthanasia? You do always use heparin, right? Is that including for neonatal?
Yes. Yeah, so we inject our mice with heparin, and then about 10 to 20 minutes later, we'll put them under isoflurane and then perform cervical dislocation once they're under. For the neonates and for any mice that's P10 and under, we do a decapitation step.
Okay.
So
do you use heparin for the neonates as well?
Yeah.
Okay. And interesting, so when you're using decapitation, I noticed that you're injecting via the descending aorta. If you're decapitating, is there not a chance that a lot of the buffers will escape via the carotid artery openings, which will have been severed when you do cervical dislocation?
So the aorta is not quite reaching the neck region. So for that, I would say that we're mainly concerned about damaging the aorta, which it remains intact.
But you enter the needle enters the descending aorta though, right?
Yeah, the cannula goes through the descending aorta.
So then what's the
And-
trick? The trick is to
And then
pull
it's,
all the way up.
yes, yeah,
I see,
exactly.
okay.
I see what you mean. Yeah. Yeah. So we can usually see the pulmonary valve to the left and that under the microscope, and that gives us a good indication of where the aortic valve is.
See, okay.
Yeah.
Great. Okay, very quick question. How long have you tried to keep your cardiomyocytes alive? I think this is particularly for the adult cardiomyocytes. Have you tried keeping them alive in just suspension, or I cultured them?
I guess we've been so focused on keeping the time as short as possible, so that's been our main focus. But we have performed calcium reintroduction steps and cell cultured them over a 72-hour period, and they looked at act and behave as you'd normally expect them to in culture. But beyond that, we haven't really tested to see how long they would last. I'm guessing that's specific to cell culture.
Yeah, my guess
yeah.
would be the quality of your cells looks so high. I think they will last as long as any other technique would
Yeah,
be
exactly. Yeah.
would be my guess. There's also a question: have you ever tried to use this to isolate atrial myocytes?
Yeah, so our buffers get through the ventricles and the atria, and then just before, for our purposes anyway, just before we collect the heart after the tissue digestion, we actually cut the atria off. And to the dismay of the atrial cardiomyocyte field, and we just collect both ventricles. So I haven't specifically tested to see what they look like, but they are digested in the process. It's just at the end we take them away and discard them. I'm sorry.
So I mean, I think this is actually an important question, because a lot of people ask me this as well. And Matt, I'm sure you've received lots of questions on this as well. What's your experience, Matt, with the injection method for atrial isolation? Have you tried it yet?
Yes, we get some cells. I don't know. I think as Amy was saying about some of the neonatal papers, it's actually very hard to get yields on how many atrial cells you're expected to retrieve from a mouse heart. We get very few, to be honest. We do get some, and you can see them. I'm talking about adult now. You can see them. They're very long and thin. From neonatal, we get you can actually get quite good atrial myocyte yields, and when you mix, I think we even discussed this in our previous webinar, when you mix the atrial cells in with the ventricle cells, you get a lot more consistent beating in culture, which we found was quite interesting. So I think maybe there's some pacemakers up in there as well. But yeah, I haven't personally worked very much on atrial cells. I couldn't tell you about their function,
Okay.
how well they are surviving or functioning after this isolation technique. But you did I think you did you do some functional studies on the atrial cells isolated by injection?
So first of all, I mean, I guess we've used Langendorf, but not we've used ex vivo cannulation, and we can isolate atrial cells from, let's say, from the left atrium. We would get maybe between 20 and 40,000 cells, so not huge yields at all, but enough to do some functional experiments. We've done some calcium contractility, and we've done a lot of patch clamping on these cells. We've also tried this using the injection method, and we found that that's not as efficient as the Langendorf perfusion. So we would get between 5,000 and maybe 20,000 cells from the left atrium. This is adult, by the way, not neonates. We've never tried neonates. But what I suspect is going on that you need the atria are sometimes very well perfused and sometimes very poorly perfused. And I don't know whether this is because of what we do, or whether this is because of the differences in anatomy of the vascular system or vascularization of the atria. I'm not sure. But we do see a lot more variability with this, with isolation of atrial cells. And that's probably the only thing I can say from our experience. But yes, you can get the cells, but Langendorf seemed to have been better for adults. the injection method, but nevertheless, you do have to vary the times a bit more, I think, and just see what works best for you. We found it to be sort of a bit hit and miss sometimes. We always get cells, but just the number of cells is a question mark.
Mm.
Hopefully that answered your question from our perspective, at least.
Yeah, so that was my experience as well. Another couple of questions, I think two of them linked. One is asking, do you use BDM? So that's, I can't remember what that stands for anymore. Butane monoxine. It's a contractility inhibitor. So do you use BDM in your media? And there's also another question here. Do you start the perfusion with a calcium containing solution or calcium free solution?
Yeah, so both good questions. We do have BDM in our diffusion buffers and our digestion buffers. And again, this is as we've taken the diffusion buffer that worked best for us from the Lau-Jan method. So yeah, it does inhibit contractility, so I wouldn't recommend it to keep it in the buffer for those types of studies. And we also have no calcium in our diffusion and digestion buffers, but we have included calcium with our digestion buffer and formed calcium reintroduction with the wash steps for the very few couple of times where we went on to culture them. But again, that wasn't the main aim for our work. So yes, you would have to add the calcium in for cell culture and functional experiments and take out the BDM.
So there's some labs who use very low calcium concentrations because some enzymes, for example, collagenase, they are calcium dependent, but I think they actually need very, very tiny amounts of calcium. And actually, I think they already have enough calcium in
from
them.
The cells.
Just from the cells they were released from, and it's not completely purified. Right? So we haven't had to add calcium for that reason. Ourselves.
Yeah, I saw that collagenase is dependent on calcium action, and then I thought, oh, we don't have any calcium. It must be residual from the cardiomyocyte. So yeah, completely agree with that.
Sorry, can I just interrupt quickly here? Because obviously some people may want to culture their cells. So with your methodology, have you tried, when you do calcium reintroduction for your culture, do you lose cells? What's the survival rate of this?
It was definitely more than 60% still, but we did see a loss of viability and cell structure as the cells became hypercontractile with the addition of the calcium in the reintroduction steps, which is one of the main reasons we took it out, so that we had more viable cells at the end. But yeah.
Fine. And Matt, I mean, have you experienced similar? Obviously, I don't know what you mainly probably do this for. You probably use isolation evenly for cell culture as well as for a sort of straight proteomics or any omic approaches that you do. So what's your experience in this?
We find that neonatal cells are very resilient. So you can put them straight into a calcium containing buffer without having to reintroduce calcium slowly, and they seem just fine with that. Adult, of course, we will introduce calcium slowly, and I think as Amy said, you'll see a slight reduction in yield over the purification steps, and if everything has worked well, that shouldn't be too much. But I think if you've had, for example, the heart was a diseased heart, you've had to work harder to dissociate that tissue, and I think you have a few more cells which are damaged. They'll have small pores in their membrane. So when you add calcium containing buffers, then those cells will be the ones which start to hypercontract very quickly and die. So yeah, it can be a measure of either how well the isolation has gone or how diseased the heart was, and you'll see a drop off in viability. What I would say is that when we put them into culture, we switched now. We used to use BDM in culture, but we use blebistatin now, which I think you you also suggested, Davor. We've done
Yes,
this
so definitely blebbistatin is a bit nicer to the electrophysiology of the cells, but obviously it's a lot more expensive. So
we have some people asking us, should we put this into our digestion buffers? I say if you're a millionaire, go ahead, but I wouldn't. BDM is just fine for the digestion buffers, but if you want to culture adult myocytes, what we usually say is at least 24, probably better 48 hours with I think we use 5 micromolar or even 1 micromolar blebbistatin. After that, you can often take it out actually, and the cells are they're a bit more tolerant at that stage. But for neonatal cells, we don't use blebbistatin or anything like that.
So keeping the BDM in the buffer is fine for downstream functional studies.
Depending on your functional study. So if you wanted to do patch clamping or contractility on immediately afterwards, you would probably have to take that out, I think.
Oh, okay. For that particular time. Okay.
Yeah.
And it's good to know about the neonatal cardiomyocytes, because I've also wondered if we were to go on to do cell culture experiments, do we need to add calcium? And then especially for the infant ages where they're a bit bigger and they may rely more on calcium than the neonates.
That's going to be the interesting one. That's for you to sort out now. So you're all of these different ages. So we know that up to about day 5 and even day 7, they seem fine without calcium reintroduction.
Yeah.
I don't think we've tried beyond that actually for culture. Have we tried any other mouse strains? This is an interesting question as well. So a lot of our work is on C57 Black 6. Have you ever tried other mouse strains?
Our lab hasn't, but another lab in our building uses this method, and they use FEV mice, and they're very happy with the protocol and published a few papers with FEVs. So we know that it works well with FEVs, but apart from those 2 strains, I'm not sure that it would be particularly different unless the structure of the parts different in terms of the formulation and the digestion of the ECM. But I suspect it would be fine for different strains.
I agree. We've tried a few strains and we don't see drastic differences that I can think of in how the cells have responded, or at least at the isolation stage, they seem quite similar to us.
Yeah.
What else do we have here? I think we've got a question. Have we ever tried to induce several damages to the heart? So this is an interesting question. Have you isolated cardiomyocytes from TAC or MI hearts, Amy?
Yeah, so our group has isolated cardiomyocytes from TAC hearts, and that really required a careful dissection to remove the surgery string so that you can actually attach the aorta to the cannula, but not for MI. We haven't tried that, but that would be really interesting to see. A lot of people have said it wouldn't work, and I question that because in the healthy tissue it would. You'd imagine it would if the coronaries for that part are still functional. I guess the next question after that is, do you need to digest the dead tissue? So yeah, I'm not sure.
Yeah, I agree. I think we have actually done our method, which I think would be the same as your method on an infarcted heart, and exactly like you say, the healthy tissue gets digested okay, and the diseased tissue is very tricky. But there's not that many cardiomyocytes in the infarct itself. But if you did want to digest the infarct itself, you probably have to chop that up, I think, and chunk digest that away. It's just Elizabeth said that they did notice differences with 2 different strains of mice, 129P2 and DBA. The mice being harder to isolate. I haven't tried those strains, but that's quite interesting.
Yeah, we've not tried those strains either in our lab.
No, we haven't.
But I don't understand why it would be so different, unless is there a different size? Sometimes different strains have different weights and sizes. I don't know whether that's the difference, and therefore you need to up your collagenase concentration or maybe even time of digestion. because of this heart size. I can't imagine the vascular system's any different.
There could be other intricacies, right? So we know that it's easier to digest, get viable myocytes from rats than mice, and one postulation for that is that there's a different resting sodium or potassium levels in the cells, which is a result of different ion channel functionalities. So if you had a strain of mouse which had more or less active sodium pumps or potassium pumps, then they might be more or less sensitive to some of the digestion techniques. I suppose that could be true. Maybe calcium related things as well, right? Because we know that these cells during digestion, they will be quite sensitive to calcium, and if you have anything which is increasing that sensitivity, you might have a lower yield, I suppose.
Yeah, I mean, it is true that the larger animals do tend to have a lower resting sodium. So mice have a slightly higher resting sodium. We've done a lot of work on this over the years, but I don't know whether that would be the reason for cell death. We've raised sodium in mice artificially through Warbane, and up to 20 sometimes millimolar, and they haven't died really. So I mean, sometimes they do, obviously, if you induce an arrhythmia, but it's they are still fairly robust. A 3 to 4%, let's say 10% increase in sodium in a mouse side shouldn't kill it off. But no,
yeah. I think the size of the heart, like you say, may contribute to different strains, being different sizes at different ages, for example.
But I don't know about these two strains, I must admit, so I'm just guessing.
Maybe on size. So a lot of the data you've shown here is P2 mice, I noticed. How small have you gone down with these pups? Have you done P0, so really freshly born pups, and even can you take this down to embryonic, or are you really reaching the limits of your aortic cannulation technique there?
So we haven't tested pre-birth. We have only tested post-birth, including the day of birth, which we can cannulate, but embryonic hearts I've not tried personally. But I would be interested to see if people can try it. Yeah,
they're tiny, because I think you guys must be very skilled. I had we were isolating P0 pups the other week, and I was having a look actually to see if I could see the the aorta coming down. I thought, wow, that's going to be really tricky. But then we don't actually use a microscope though, so I think that's probably a difference if you have it on.
Yeah, you have to get used to the anatomy, and also I'd say even P0 to P2, the aorta is kind of like jelly at the P0 stage, and so even those 2 days help to keep it intact, because when you're learning, you will accidentally grip the aorta too tightly and it will tear. And that's actually another reason why it's nice having that longer section, because if it does tear where you've pinched it, you can kind of just cut that bit off and then start again, start your cannulation again. So that's a good bit about having the longer section. But there's so many tips and tricks in this field, and it is difficult to get all that information out there, but also to find that information. So hopefully it helps people.
So I guess contact Amy and Matt for all the tips and tricks. Sorry, you're going to kill me. You may get inundated with emails.
All the good bits we don't tell you in there. Are you doing this yourself, Amy, when you're down there isolating cells, or is it like a 2-person operation?
So it's
1 person.
Yeah, so we actually had a really technically gifted RA who was able to do these cannulations, and I did the cannulations along with her. But what we normally do is tag team. So she would do the Langendorf fusion, and I would do the rest of the process. And if she wasn't around, I would do the cannulations, and then there was another RA who could do the isolation, the purification process. So between the 3 of us, it made the whole day go quite smoothly. But she would simultaneously cannulate two hearts at the same time using 2 rigs herself. So it is possible to do by one person. I've done that myself as well. So the hearts that you saw in this presentation are the ones that I had cannulated. But yeah, so once you've got one heart attached to the rig and press start to let the buffers flow through, you can get started with the second heart. And by the time you've taken the first heart off, because it's only 4 or 5 minutes with the cannulation, the second one's ready to be taken off as well.
Great. I think we're at the end of our formal questions. It's a mammoth effort. Thank you very much, Amy. Is there anything else you'd like to? Any other tips and tricks or anything you want to divulge now, or is it? Yeah, is that enough for a day?
I think that's everything from my end. I mean, it's all out there now, so hopefully it does help. But definitely seeing previous papers and yields and your paper and all the Langendorf papers and the batch chopping, all of those have contributed to where we are today. So I would never say this is better than this. It's just a development. And I guess we're all trying to improve it for our specific uses. So for a functional study, this might not be for that purpose, but for our purpose of getting good molecular characterization and morphology, it's been really handy.
Guys, if that is the case, I think we've run for an hour and a half, which I think is impressive. So, Amy, well done. And Matt, thank you again.
Thank you.
The one thing I would just do before we wrap up is just share one more time the next webinar slide. And the reason for sharing this is because I've now got a shorter URL link, and equally there is a QR code as well for anybody that wants to register for this webinar. But on that note as well, again, one more time, Amy, thank you so much for writing a really, really nice paper, really nice study, and for spending so much time with us. Matt, thanks always as ever for constant advice, support on this topic. Your knowledge is unbelievably useful for everyone. May I just say that, if anybody wants to contact anyone here, please do. I know Amy and Matt will kill me, but I definitely know Matt's been responding to lots and lots of questions, and I'm pretty sure Amy will be soon as well on isolation. So thanks again, everyone. And see you for the next webinar, hopefully.
Thank you very much, Davor.
Yeah, thank you so much. It's been great.
Bye, everyone.
Yeah.
Bye.